Hello everyone, welcome to my session. So, in this session we are continuing with the same topic, ocular drug delivery system. In last two sessions we have discussed about basic introduction of ocular drug delivery system and the formulation considerations of ocular drug delivery systems, various barriers involved in ocular absorption and how to overcome these absorption and alternate approaches to use for better absorption of drug, right. So, now in this session we are going to discuss about different type of dosage form we have for ocular administration. Though we have discussed slightly about conventional dosage form in last session, but now we will again going to discuss all the variety of formulation available for ocular administration in this session as well, so that it can be revised for you. Now, without wasting much time, we will move on to next slide, that is novel ocular drug delivery systems. So, what are the various objectives for making all these novel type of formulations? So, here I have listed few, right. First is to increase accurate dosing. As you know, the problem with conventional ocular drug delivery systems are, yes, it may eliminate out while instillation or during instillating into the eye, the majority of drug is getting lost. So, this results in the poor bioavailability of the drug, and the poor low percent of the drug can be passed through the corneal or the eye barriers to reach the ocular region, right. So, with these problems we need to develop an alternate approach for better performance of drug candidate within the eye. So, first is to increase the accurate dosing. Yes, in conventional types of form there is loss of the drug during instillation of dose. So, if we are talking about novel drug delivery system, then the dosing accuracy is of important concern. To overcome side effects of conventional system, right. These are the side effects which I have told you, that lacrimal drainage, tear fluid dilutions and your nasolacrimal drainage, right. So, all these are the problem associated with conventional, though the route of absorption or the elimination part is same for all type of preparation going through eye, but it can be controlled or it can be manipulated in that way that we can have the maximum results for drug within the eye region. To achieve sustained and controlled drug delivery, right. Yes, if we made these novel type of preparations, we can have different profiles of drug in terms of controlled manner, sustained manner and extended release, right. To increase the ocular bioavailability of drug by increasing the corneal contact time. Yes, retention is more of drug within the eye region, then it will automatically improves the drug absorption as well as the bioavailability of drug in particular region, right. To provide drug targeting within the ocular globe, so that the loss to other ocular tissue can be prevented. Yes, drug targeting is the main aim of making these type of preparations for ocular region. Yes, it prevents the loss of drug to other parts of the eye, as it is making for the targeting effect. To overcome the protective barriers like drainage, lacrimation and conjunctival absorption. So, barrier properties we have discussed earlier in the sessions. Yes, so we can overcome these type of problem associated with physiological barriers of eye, right. To provide comfort, improve patient compliance and therapeutic performance of drug. Yes, to provide comfort to the patient and improve patient compliance and therapeutic performance of drugs. So, overall these things can be achieved while making these novel drug delivery system. Now, move on to next slide. We will discuss about the formulations including conventional, alternate to conventional, like different type of injections we have discussed, right, and then the novel delivery systems. Now, first we will talk about the conventional. Includes eye drop, the mostly used preparation and mostly accepted preparations we can say in the market. So, administered as solution, emulsion and suspensions. The retention is affected by the properties like hydrogen ion concentration, osmolality, viscosity and instilled volume, right. Low absorption is achieved through these type of preparations, approximately 5%. You remember I have shown you a chart which represent the ocular absorption 5% and remaining is getting absorbed in the systemic circulation. So, 50 to 100% varying depending on the types of drug we are using. So, with this the only 5% drug is available to the eye region which shows effect. The dose is mostly absorbed via the conjunctival and nasal blood vessel, that means nasolacrimal route. The drainage is happened of when instilled as an eye drop. Now, the next type is ointment and gel, that is semisolids. So, semisolids are useful for prolongation of drug contact time and within the external ocular surface. Yes, you put the gel preparation, you have seen that a variety of gel preparations are available in the market for different type of disease condition in the eye, right. So, the patient in diseased stage or altered eye physiological factors may apply gel preparation on the eye surface, but may feel some sort of discomfort as the vision is blurred. The major drawback of this dosage form like blurring of vision, matting of eyelids and which limits its use, though the retention of drug is high as compared to your liquid preparations, but due to this comfort of the patient, this limits its use in the patients. So, in disease condition already a patient is suffering with so many of discomfort feeling or discomforts. So, after having these type of additional discomfort of blurry vision and all, so patient may feel disinterested completely for these type of preparations. So, very in rare cases or in very less cases the gel preparations are preferred or prescribed to a patient until and unless it is very important. Vesicular system. So, now after discussing about the conventional forms, we will discuss about the novel drug delivery systems like vesicular system, liposomes, niosomes and discosomes. So, in vesicular system we have variety of systems like liposome. What are liposomes? These are bilayered vesicles which are biodegradable, non-toxic and amphiphilic systems, right. So, liposomes are made up of phospholipids, right. These are bilayered consisting the bilayer vesicle structure, right, and is made up of phospholipids, right. So, these are biodegradable because it is made up of natural component. So, these are utilized within the system and they do not harm as it is have similar nature as your body is made up of, right, body layers is made up of basically. So, these are non-toxic, amphiphilic, delivery system consists of phospholipids and cholesterol, offers better permeability of entrapped drug, right. So, they can easily absorb through the eye layers and gives better absorption of drug. Sustained release of drug into vitreous and retina-choroid compartments can be achieved using these type of liposomes and avoids non-targeted tissue. They can have targeted type of delivery or delivery of the drug candidate, and they can avoid the non-targeted tissue like lens and sclera directly reach to the particular site. Now, we will talk about niosomes and discosomes. So, basically niosome and discosomes are the another category of liposomes in which instead of phospholipids, non-ionic surfactants are used, right. So, along with its cholesterol part. So, these are bilayer structure similar to liposome, which can entrap both type of lipophilic and hydrophilic drugs, and it exhibits low toxicity and more stable as compared to liposomes. See, it has synthetic type of non-ionic surfactant component, and in liposomes phospholipids are there. Then stability issues are there with the liposomes. So, it can be overcome using niosomes and discosomes, as it has non-ionic surfactant component along with cholesterol and can be used. In discosomes, the surfactant used to make discosomes cellulin C24 has been used. That means, the shape is like disc. So, it is named as discosomes. The size of discosomes varies from 12 to 16 micrometers, favors these system not to pass to the systemic circulation. So, this is basically the advantages of making and using these type of delivery systems for ocular administration, as you may have the targeted type of delivery of the drug candidate. Now, after vesicular, we will discuss about controlled delivery systems for eye, right. So, controlled delivery systems for eye is first is implant. Though in your syllabus implantable drug delivery system is an separate topic which needs to be discussed in detail, right. But in ocular administration, implants are also has an important role. So, we will highlight this topic not so in detail, but slightly discuss this. So, for specifically for ocular administration. So, controlled delivery system first is implant. You see in the picture that variety of implants have been used. Now, it is effective for cytomegalovirus retinitis. This is the disease condition in which these implants are indicated, and or we can say in these type of disease condition only implants are indicated to cure the condition well as compared to the conventional one. So, these are non biodegradable polymers need surgery to remove after the treatment. Yes, these implants for CMV retinitis condition are made up of non biodegradable polymer. That means, they need to be removed after the treatment or therapy. Biodegradable polymers can be used to make these type of preparation, that are a polylactic acid PLA, are safe and effective to deliver drug in vitreous cavity and show no toxic signs. So, these are different biodegradable and non biodegradable polymers can be used for implants, right. And you see that in the eye region, yes, this is the device through which we can put implant into the eye region. Now, you see this is the picture where in original you see this highlighted area. This is the implant which is put on the eye, right. So, here is the region which is put on the eye using this device. So, now you see how implant is incorporated to the eye. Now, next, ocular iontophoresis. This is again a different approach to give the drug to the ocular administration, to give the drug to ocular system for controlled delivery, right. So, direct current drives charged molecule of the drug ions into cell or tissue, right. So, it is the device which is put on the eye to current is involved in delivering the drug actives to the eye surfaces. So, you see in the picture, positively charged drug are driven into tissue at the anode and vice versa. It is fast, painless and safe, but it can also deliver high concentration of drug to a specific site. Now, I will brief about the picture. You see, this is the device, it has two types of electrodes, anode and cathode, and this device is here, and this is device is put on the head of the patient, and this is on the eye of the patient. This wire or electrodes is put on the eye ball of the patient, and now you see, after putting those devices on the patient, this is capsule which is placed on the eye, and it is delivering, after giving the charge to the through the device, it is now getting, the drug is getting discharged through the eye wall, right. So, this is how you can have the controlled release drug through iontophoresis. It's not very widely used, but it can be used for controlled drug delivery of the drug candidate. Next is dendrimers. Dendrimers, these are tree-like nano-structured polymers. Again, these are nano-structured polymers, that means nano carriers. So, and have better water solubility, bioavailability and biocompatibility. So, these are basically nano-structured carriers which have better permeation, and again they can be targeted to the eye region, required region. They are attractive system because ocular drug delivery due to nano size range, ability to display multiple surface group that allow for targeting and easy preparation and functionalization. So, you see, these are nano structured carriers which can be targeted well within the eye, and you see the structure, these are branching points and these are the core generation numbers. So, the structure of dendrimers is like this only. Fourth one is microemulsion. So, microemulsion, again the micro reach vesicles are present. So, you see the size is between 5 to 200 nanometers, and which may be varied depending on the type of drug you are using. Thermodynamically stable phase transition systems, also known as, which possesses low surface tension and small droplet size, offer high drug absorption and permeation, and provides drug delivery to posterior segment of eye. Posterior segment, this the outer side is considered as anterior segment, and the back side of the eyeball is considered as posterior segment. So, basically these microemulsions target the posterior side of the eye, right. Nano suspensions, efficient delivery of hydrophobic drug and enhanced rate and extent of ophthalmic drug absorption. Using these nano suspensions, we can have maximum ophthalmic drug retention within the eye region where we want the drug to be retained, right. The intensity of drug action with significant extended duration of drug effect. So, while drug is retained within the required region or target at the target site. So, we can have the extended profile of drug in that region after having these nano suspension as drug delivery system. Next is microneedles. Microneedles again an advancement in ocular administration of drug. You see in the picture that these are microneedles. This shape considered as the microneedle thing and self-adhesive microneedle patch. So, it is available as self-adhesive microneedle patch, a type of patch which has minute projections or outer projections which can be placed on the eye in a particular region, and after inserting this patch on the eye, it will start releasing the drug in sustained or controlled manner, right. Minimally invasive and feasible vehicle for sustained drug delivery. Delivered drug into sclera, that is posterior segment again, to treat diseases and avoids the complication associated with intraocular injection and systemic administration, right. So, microneedles can be a good option for those who to overcome side effects of intraocular injections and systemic administration of drug, to surpass these type of side effects. Now, you see in the picture, after putting these microneedle patch on the required region, that is your sclera posterior segment, it is anterior segment, you see it is anterior segment and this is comes under posterior segment of the eye. Your patch is put here and these are outer projections. Now, you see these are considered as microneedles. So, after inserting into the eye region, required region, the swelling is now can be seen. So, it will make this needle swelled state and after this interlocking features. So, once it is get swelled, then it will start releasing the drug to the particular region at the target site. Now, I hope you understand this point. The patch contains the minute outer projections, and after inserting or placing the patch on the posterior segment of eye, the inserted microneedles will get swelled after absorbing the the environmental moisture and then it gets swelled and will start releasing drug molecules. Now, the seventh one is collagen shields. Collagen shields are disposable, cross-linked, short-term therapeutic bandages lenses for cornea that conforms to shape of the eye. That means, these shields are flexible in nature and they can be fixed anywhere in the area and they can form any shape while retaining in the eye, right. Shields are sterilized by gamma radiation. Irradiation delivers drug and promote corneal epithelium healing. Soaked in water-soluble antibiotics and steroid solution prior to placement and biocompatible. That means, these are non-medicated. If you want to apply the medication through these collagen shields, you need to soak it with the solution in suspension, medicated solution in suspensions, and then once it is filled with soaked properly, you will apply on the eye region. Now, it causes discomfort and interference with the vision. So, that is why it is not preferable, because it may have vision problems, right, like this. This picture represent the collagen shield, how it is look like. Now, if you see the eye, this is the collagen shield where it is applied. So, basically collagen due to sorry, this is not the shield, this is abrasion and this is the shield applied on the eye. So, this collagen shield when applied to the eye or lens, so we see that some sort of abrasion on the eye surface can be occurred in patients. So, they may cause, it may cause discomfort, and this is the actual picture where you see the shield is applied, and this is the aberration part where some sort of reflection and friction can be seen in patients. So, this is not so much acceptable because it already the patient is in not unwell stage, and again it is causing inflammation or irritation in the eye, then the patient acceptability is less in that case. Now, contact lenses. So, contact lenses, widely used lenses or small layers of. So, ocular contact lenses offers correction of refractive errors of eye. So, instead of spectacles, many people use contact lenses for correction of refractive eye. So, medicated lenses are also available in the market. Therapeutic lenses are often used to aid corneal wound healing in case of infection prolonged manner, right. So, therapeutic lenses or medicated lenses are also available. Increase residence time of drug in ocular region. Yes, when you put lenses on the eye surface, then it is in contact with the eye surface for long time, and it is release the drug for prolonged time. Now, for example, Bionite by Griffin Laboratories Bionite is a medicated contact lenses manufactured by Griffin Laboratories, and soft lens are the lenses prepared by Bosch and Lombs. So, you see the picture here, the a person is applying contact lens. So, proper SOPs are available how to apply contact lenses at if it is not placed properly, it may cause irritation to the eye. Next is mucoadhesive system. So, macromolecular hydrochlorides having hydrophilic functional groups like hydroxyl, carboxyl, amide and sulfates involve in electrostatic interaction. That means, mucoadhesive approach has also been used for ocular administration, like if we use this approach, the eye and the dosage form retained on for more time on the eye surface, and it will release the drug for prolonged time, right. So, you see the form dosage here, you see the dosage form, and which has the mucoadhesive polymer, and it is making bond with the eye surface. So, we have discussed in mucoadhesive drug delivery system that the interaction is taking place in the consolidation part of the stage of the mucoadhesion. First initially contact and then consolidation. So, this is how the drug is adhered to the mucosal surface. 10th one is ocucerds and lacricerds. Ocucerds here, it is the it is placed within the this region, or which is known as cul-de-sac. Ocular inserts is a sterile thin multi-layer drug impregnated solid semi-solid consistency devices placed into conjunctival sac, or also known as cul-de-sac. Lacricerds is a sterile rod shaped device to treat dry eye syndrome. So, where these are the different inserts for eye preparation. Next, microparticles and nanoparticles. I have told that these solid particles in being in micro nano range are the suitable drug delivery system for ocular administration. Here are the references, and I hope you understand different forms of ocular preparations we can use as novel drug delivery system. And the next session we will be discussing about the evaluation part of these preparations. Thank you, stay safe, take care.